getting himself configured there. I'll just give a brief introduction today, and I'll give a more extensive one tomorrow at our more formal colloquium. So I don't know if all of you have already met him. This is Professor Ellery Ingall from Georgia Tech in Atlanta. And he actually, I know him from many years ago through common graduate student origin at Yale University. And Ellery was a student, and I guess still is intellectually a student of Robert Werner, who was, of course, a renowned professor at Yale for many decades, working on biogeochemical processes and sedimentary geochemistry. And Ellery is very well known for his work in phosphorus chemistry and biogeochemistry of phosphorus. So tomorrow he'll actually be talking about penguins, but today, right?
Yes.
Isn't that true?
Diatoms and penguins, yes.
Sort of. That's a joke, which you'll see tomorrow. Why it's a joke. One of my lousy jokes. In any case, he has been at Georgia for about 15 years or so. Before that, he was at UT in Austin, and also did postdoctoral work at Skidaway, which must have been a nice adventure. And that's obviously what attracted him to go back to Georgia, his experience at Skidaway.
Nice place.
Yeah. And so today we're lucky Josie cajoled him, in addition to giving the regular colloquium, and giving this special seminar today on the topic of aerosol chemistry. And so there's the title, and Ellery,
thank you.
A better introduction tomorrow.
Thank you so much. Well, one thing, since we're a nice small group here, if you have questions and you just want to interrupt while I'm going, please do. I think that just makes it a lot more fun and more lively. It's a nice mood lighting there. There. So, going to look at aerosols today, and this has been, I hate to use the term, but I've been just having a whole lot of fun over the last few years applying synchrotron techniques to looking at the chemistry of atmospheric aerosols. And there's many reasons to look at aerosols, both in urban settings, which we've done in terms of pollution, the solubility of metals in urban aerosols, factors into the formation of reactive oxygen species. So we have that aspect of work. There's sources and source apportionment on the urban setting. But of course, there's also the greater oceanographic reason to look at metals and aerosols, because they can supply things like phosphorus and iron to ocean regions that are in sore need of this to support productivity. So this fun has been, the synchrotron techniques, which I don't need, you've got one right down the road here. They're really good at looking at small things. And aerosols are small things. So that's been fun. And one of the things that's always struck me funny is, a few years ago when we started doing this, it was like, wow, there's just not a very abundant literature at all. There's some, but not as much as you would think. It's a really nice way or different way of looking at aerosol chemistry and characterizing it. But it's just not it's a tremendous amount of work done. It seems like the soil people have jumped on the synchrotron work, and some of them, marine particle people, like a dog on a bone. But the aerosol people seem to be that. So we're going to look at, we've looked at a lot of elements, and they're all like precious babies to me. But I'll just try to focus on phosphorus and iron today, two major marine nutrients, and more oceanographically oriented studies. Very important. I put her picture on the slide. This work has really been driven by one of my really awesome graduate students, Amelia Longo. And the phosphorus work we published a couple years ago in GRL. And we have the iron work, which I'll present today, that's in review right now in ES&T. And of course, I have all sorts of just wonderful, that's one of the fun parts of this work, is all the great people that you get to interact with. So I have to just start out with a silly story. So we got all started on this aerosol work. It's those of you in the faculty world, you get assigned to these jobs, and you're, so, I was on a comprehensive exam committee of this student, Michelle Oakes, and, you're thinking, oh, I got to give this stupid exam, and this will take time away from, like, surfing the internet to get the latest status of Kim Kardashian's posterior and other important things. So, but, Michelle was, giving a wonderful talk. She was studying metals in Atlanta Aerosol and had developed some online measurement techniques and such to better measure, in particular, iron in Atlanta Aerosols. And somewhere in her talk, she said, we're really worried about solubility, but, we just don't, we can do the liquid phase, different oxidation states of iron, but we really don't have a way of characterizing the oxidation state of iron easily in aerosols in the solids. And I'm like, I think we might be able to do that. So we were able to sort of, I talked to my Synchrotron guy, Barry Lai, at Argonne National Labs. I said, hey, can we give this a try to see if we can get decent signals on our Atlanta aerosol samples? So we first just went out and got some test samples to see if it would work. And one of their, there's a whole monitoring program in Atlanta. One of the monitoring sites is near Fire Station 8. And so we're getting some filters and such to test. We're thinking, it might also be nice to get some source materials. Like, oh, but what would, I wonder if we could measure diesel exhaust. So Michelle walks over to the fire station next door to the sampling site and, can we borrow your fire truck? And as you can see, they're very nice, and we set up our sampler. And indeed, we were able to get signals. For our real published work, we got official EPA diesel exhaust samples, blah, 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 with all the protocol. But this at least gave us a window that things work. So I just think it's just kind of fun thinking about that. So at a marine science and aerosol science place, I think we can just stipulate that phosphorus and iron are really key nutrients in marine systems. One of the systems we'll be looking at today is the Eastern Mediterranean, which is thought to be phosphorus limited. Production is phosphorus limited there. And of course, many regions in the Atlantic are thought to be at least iron limited at times, if not for extensive periods of time. And so some of the things we'd like to address is, okay, using these synchrotron techniques, what kind of differences do we see in the composition of different source regions? So I can give you, at the beginning of the talk, in terms of phosphorus, the composition of phosphorus depends on which way the wind blows. So that's one thing I hope to show you today. And then when we look at iron, what's happening as that aerosol is moving through the atmosphere? And so for this study, we looked at aerosols exclusively originating in the Sahara Desert, where they start out mostly as iron oxides, and looked at them in different sampling locations to see what happens. And to just give you the result that I hope to show you today, the iron in that aerosol gets more reduced the longer it's in the atmosphere, and we see different phases, iron phases coming into those aerosols presumably forming during transport. So there's a lot of action in this aerosol as it's moving around. And I think that's kind of fun. And we can speculate, well, what's happening? Photoreduction, proton reactions, while these aerosols are moving from one spot to another in the atmosphere. So for the phosphorus study, we're going to stick to Eastern Mediterranean. We have some colleagues in Greece, and they have a sampling station located nicely on the Isle of Crete. And this is a good spot because sometimes the wind blows from Europe, and sometimes it blows from North Africa. And of course, we can select samples where the wind was essentially exclusively coming from the north or from the south for our samples to get an idea of compositional variations from two different source regions. So for aerosol work, and probably for various obvious reasons, the Sahara gets all the attention. You know, and it's not hard to go on the internet and find about 1 million satellite views showing big dust plumes moving across the Sahara. Here's the Nile Delta. If you don't recognize the area, Nile Delta here, and the Isle of Crete is under the clouds here. But big plumes of dust. Certainly that's got to be a big source of potential materials, nutrients like iron and phosphorus to the Mediterranean. So, but we're going to also look at the material coming from Europe. We did all of our work on this project at the Argonne National Laboratory Synchrotron. For the phosphorus, we did all our work on the 2IVB beamline, which is a very unique, somewhat lower energy beamline. It's one of just a few in the world that you're able to do phosphorus microscopy and spectroscopy. Unfortunately, it's being torn apart. So hopefully they're going to build one at Brookhaven pretty soon here, but I think that's a couple years away. But it's a loss for a really nice resource. And then we've done the other work on a adjacent beamline that's a higher energy. We can look at other elements. So one of the things that as we've done this aerosol work is how do we approach these aerosols? So essentially, these aerosols are size fractionated with various sampling devices into essentially a 2.5 micron and smaller fraction, sometimes a 10 micron and smaller fraction with a sampling device, and just deposited on a filter, usually Teflon or something similar. And what we found is we're trying to extract every last little bit of information that we can get from the synchrotron on these aerosol samples. So our approach has been to take a piece of filter. I'll just show you the here's the sample mount. So this is just aluminum stick with a little slot in it right here. And you mount the filter over the slot. So that is the sample prep. That's one thing I really like. The other way, of course, of classically looking at aerosols and aerosol samples is putting the filter in some sort of solution, doing some sort of sequential extraction, which we've learned a lot from, but it's also really nice to just be able to put a piece of filter over a little slot. And we can, first, one thing we've done is we'll just use a very focused beam, raster the beam across an area of filter, and create a map. And what we've noticed is when in either iron or phosphorus is that there are certain particles that stand out as phosphorus-rich or iron-rich in this sort of matrix of usually a little bit finer material. So you'll see these 1 and 2 micron sized particles that are enriched in these elements. And in some studies we've noticed that the composition of the large particles, well, let me get to the next thing. So we can identify these particles and then go back to them once we have a map, we know where they are, put the focus beam on the particle, vary the energy of the beam to do various forms of iron or phosphorus, X-ray absorption, fluorescence spectroscopy. So we vary the beam, and the spectroscopic patterns give us some idea of the chemical form of the phosphorus or iron under the beam. So we can do individual particles, which we can locate in a map. And then we realized, well, it'd be just nice to get the general spectroscopy of a whole filter. So we hit the button that removes the X-ray focusing lens, called the zone plate, moves it out of the system, and we can move a fair bit of the X-ray optics out, blast a fairly large area of filter with a beam that's a couple square millimeters, which is big in the synchrotron world. Normally our beam for phosphorus is about 100 nanometers spot, and for iron it's about 300 nanometers. So very finely resolved. And then so we can do this bulk spectroscopy, and sometimes we see that the individual particles are different in composition than the bulk, often more reduced, or sometimes they're exactly the same. So I think we get some clues by comparing what are the bigger particles and that we can individually characterize versus the whole sample. And then another little piece of information, we could say, we've got some particles here that are enriched in phosphorus and iron. We can essentially do an incredibly fine scale map of one individual particle and, depending on the energy, give you an idea of what other elements are associated with the iron or the phosphorus. So here's just an example of an Xray map. Here's a 10 micron scale bar, so it's about a 40 by 40 micron area filter. So you see all the little spots. The brighter colors are, of course, higher concentrations of iron, and in this case we have sulfur. And sometimes we can do a series. Essentially, we can get maps of every element below iron and atomic mass down in the periodic table too about sulfur. And so we can create elemental maps, but we can then focus in on this particle with and do maybe a 3 micron by 3 micron map with very small steps, the diameter of the beam, to specifically characterize the different elements within a specific particle. And then this is an example. We could find a particle like this. We vary the beam energy. We get a absorption, X-ray absorption spectra. This feature here is called the K-edge. And these very subtle bumps and wiggles give us ideas of little small things, but they're very diagnostic of what is there. So we did just recently, this is a little bit of a tangent, but it was like a technical challenge for us, is we were looking at, in another study, sulfur. And just to give you an example of an individual particle map, and we developed some technique where here's an individual particle, and we mapped it at different X-ray energies. And basically, if you map it at a high energy, all forms of sulfur light up. So sulfur in the +6 oxidation state, +4, 0, -2. They all light up. So we can map where all the sulfur is in a particle. But if we use a lower energy, essentially we can just define the distribution of just where the sulfur 0, and if there's sulfur -2 in the particle. So here is a map of the same particle. Here is the distribution of essentially sulfur 6, or all forms of sulfur, in the orange. The bright colors are the highest concentrations. And here's the distribution of the sulfur 0, and these are aerosol particles from Atlanta. And you can see here there's essentially a little glob of sulfur 0 in this particle. Here's another particle. So we're down to the scale of, with a 100 nanometer beam, of even being able to, in this case, create maps of oxidation state within a single particle. We can also do a map of composition. So this has been lots of fun for us. Okay, that was my digression, but I just am excited.
How do you store the sample to prevent alteration of oxidation state in these fairly reactors?
We've been mostly just keeping them frozen. So yeah, it would be nicer to be able to preserve them better. But every sample we've run pretty much now has been just colleagues from Greece, Bill Landing with the GeoTraces program has been very generous with samples taken on shipboard, which I'll show you, and samples from Bermuda. So mostly we're stuck with frozen. Although we find that that is a concern, obviously, oxidation, and essentially hitting it with a high-power X-ray beam is a nice way to oxidize the sample. But what we found, we've blasted an individual particle many times, just with the beam for quite a long time, and they seem to be pretty stable. The one thing that's a killer is moisture. So if there's any moisture there, hit it with the X-ray beam, you make hydroxyl radical, and there was one time we were looking at diatoms, and they weren't quite fully dried, and we were like, wow, every diatom had this circular feature in the center of it. Well, we drilled a hole in it with the beam because there was some moisture there. And so lessons learned. Okay, so here is one of the bottom line figures for looking at phosphorus in the island of Crete. When we look at European sourced aerosols, which we know the sampling date, we can do high split back trajectories to say where was the air mass the day before it was sampled, where is it sampling, where was it 3 days before. So that's good. The other clue, but it's not nearly as scientific, is you can look at the filters, and not surprisingly, the filters that have an origin in the Sahara are reddish compared to the European ones, which are kind of a gray-brown. So here is the solid line is a X-ray absorption spectra for phosphorus for North African sourced aerosols. And we have these distinctive, in the synchrotron peaks in the spectra, a fairly wide central peak, which basically tells us this is going to have a big component of apatite. We'll get to that. We can take a database of standard materials, and we've created very large databases of mineral, every phosphorus-containing mineral I can find. I've talked to a lot of really kind of strange mineral dealer people, and bought a lot of minerals to make a nice database. And so we can fit a database of standards, a linear combination fit to get the best fit for our natural spectra. And here's the fit that we obtained for the best combination of all our standards to fit this unknown spectra. And here is the European sourced aerosols, a nice average spectra. And again, here's the linear combination fit in the dashes. And there's, in the synchrotron business, these spectra are extremely different. And that's borne out by the results of our linear combination fits. This is just a little bit of an advertisement. So we put a lot of effort into creating this database of standards. So we started doing this, there was not a huge amount of literature, especially in phosphorus, and the frustration would be there'd be this study, it'd be maybe a soil study, an agricultural study, and everybody would run their own sets of standards for their study so they could interpret their synchrotron stuff, but there was no data available, so we just had the idea of running, these are, I know you can't read the names, but just different phosphorus minerals, have many more, and I just have a smattering here to show you the position of various peaks in these spectra above the absorption edge. They do move around. They are diagnostic for different mineral forms of phosphorus. So we published a lot of the phosphorus data in just a paper on standards, probably the most boring paper I've ever written. And but all the data is there as supplemental material. It's already been assimilated by one of the synchrotron people at Stanford, Sam Webb, and put into his 6-pack modeling program. But it's there for people to use, and it's been nice. I've been seeing a lot of people actually using the data now rather than wasting precious synchrotron time rerunning standards. And all these I characterized independently with XRD before running. And we've continued that trend. There is more data out there for iron, but we've also published all our standard data supplements in Michelle Oak's paper, and more phosphorus data in Amelia Longo's paper, again, as supplement. So it's out there so people don't have to keep doing this stuff. But all those standards allow us to do these linear combination fits to get an idea of what different phases contribute to the composition of phosphorus in aerosols. So we have, let's number about 10 samples of North African source aerosols taken on different days in this Crete sampling site. And this is the results of our linear combination fitting, showing the abundance of different phases, with apatite being red. We've grouped alkaline and alkali metal phosphates in green. We can't really tell polyphosphate, sort of a linear chain of phosphate atoms, from organic phosphorus. Organic phosphorus is just, the spectra are just not that interesting. They're fairly featureless, which is unfortunate because we'd love to be able to do more with that. And then metal phosphates in purple. And then here's the European aerosols, again, polyphosphate and organic P in blue, metal phosphates. So Europe over here, Africa over here, and I think that's just in a colorful way showing you that the composition is very different. So the composition of phosphorus depends on which way the wind blows, with the Europe-sourced aerosols being really dominated by organic forms, organic or polyphosphate forms of phosphorus, and certainly a lot more mineral forms are coming from North Africa. We can also, I did mention, here's a map, one of these maps that we can do of rastering the beam. These bright points are what we would call phosphorus hot spots, so particles that are rich in phosphorus. I think this is an African aerosol. These maps cover the same area. They're just a little bit smaller, but we could overlay them and could blow them up. But to show you, is silica co-located, and we can do a lot of extra work looking at elemental co-location and such. But for phosphorus, basically, the composition of the large particles we see in the African aerosols, they're almost always apatite. And for the European aerosols, the larger particles that we can resolve, they look like organic Ps. So in this case, the large particles match the ground mass. So what is the consequence for this difference in composition for something that we care about? The solubility of phosphorus in these aerosols. Because it's the ultimate solubility. When you lay that aerosol down or it gets deposited in the Eastern Mediterranean, if the phosphorus is not soluble, it won't stimulate productivity. And so here we have total phosphorus content, micrograms P per milligram sample on the filter, the soluble fraction, and we've just ratioed the soluble fraction in this dark line here with the spots. And in general, what you can say is that even though the European aerosols mostly contain less total phosphorus than the African aerosols, as these open bars, the phosphorus that's there is about 3.5 times on average more soluble than the African aerosol. So even though there's these beautiful plumes you could see from satellites, poor Europe should get a little credit for maybe being a nice contributor of phosphorus to the Eastern Mediterranean. And as you might expect, we try to, can we correlate the solubility to any one particular phase that we've identified in the aerosol? And the answer is, well, not really. There's a slight correlation, R squared of 0.6, if we look at the fraction of soluble P versus the percentage of organic phosphorus and polyphosphate. But there's obviously a lot of factors contributing to the solubility, and just that these are so rich in organic P is probably why we're getting a little bit of a correlation there, but it's not something I'd really want to write home about.
Have you looked at the aerosols in Northern Europe, or would they be the same if you were saying looking at the Baltic?
Oh, that's a really good question. Actually, it does seem like there's a compositional difference whether they're coming from Western or Eastern Europe. So, but we just didn't have enough data richness to, and so our Greek colleagues are collecting more samples for us to, and so we can maybe start to discriminate. But yeah, there is, and I think you just think of maybe a little bit more pollution contribution in Eastern Europe versus Western Europe, and that would have consequences for secondary reactions and such that could increase the solubility. So this organic phosphorus, so there's no perfect way of characterizing aerosols. So the frustration, of course, is when you're doing chemical extractions, they're always operational. And so you can learn a little bit what's there, but you have to trust that your operational extractions are okay. And the synchrotron has different problems, but I think in combination we can start to learn a little bit more about what's there. The other thing we've done as part of our work that started out with borrowing a fire truck and then got a little bit more scientifically rigorous is we've gathered up aerosols produced by a number of different common anthropogenic sources. So coal fly ash, we have a couple very large electric generation plants around Atlanta that are coal-fired, diesel emissions, volcanic ash, not a lot of volcanoes around Atlanta, but nevertheless, pollen, couple of bacterial isolates that I bought years ago. They don't sell them anymore, from Sigma Scientific, Bacillus subtilis, and a soil bacteria, Azotobacter vinlandii, and then biomass burning ash, and the aerosols produced during biomass burning, and gasoline. So these are all things that we have these aerosol sources. We've characterized them for phosphorus, and we've looked at them for iron and selenium and sulfur. So we've been using these source materials. So we had a question, okay, well, we have my mineral database and database of standard organic compounds that we use to fit our aerosols, from Europe or Africa. But what if we use these source materials as the data to try to fit those spectra? And they actually don't fit quite as well. The thing that fits the best, especially in the European aerosol, if we are going to use these source materials, is bacteria. So that's, and I know folks here have been thinking about transport of bacteria and coming from seawater, and I think the case here, but it's interesting, and I know this is something that is a good topic now in the aerosol community, is what is this contribution of aerosols to the movement of bacteria? And I think some of the other data that we see in this and other elements are suggesting, maybe we should look a little closer at that possibility. But bottom line for phosphorus, European aerosols are delivering a lot more soluble, 3.5 times more soluble phosphorus. We have compositional difference related to source region, with the European being dominated by organic phosphorus. Ultimate source of phosphorus, especially when it doesn't fit any of the common emission sources, which we certainly wouldn't expect much of this from the Saharan desert, but these don't fit too well in the European ones, but other than this bacterially associated phosphorus. So that's the phosphorus story. We see a compositional difference. With iron now, we are going to look at what happens to iron as it's moving through the atmosphere, and we're going to look for evidence for reactions like photoreduction and a proton production or acid reactions. So again, we're going to go back to our site at the Isle of Crete here, but in this case, we're only going to look at aerosols sourced in the Sahara. So we have the Crete site, and then this is West Africa here. And you can see, again, I think these are the Cape Verde Islands here, and very well-known plumes of dust blowing off the Sahara from West Africa over the Atlantic. I was once on a cruise out here. I don't know if people have been on the Nore, and it seems like they have the intake for, I think, the ship's air conditioning system wisely located in the lab. And so, but the fascinating thing is they change the air filters while we're on the cruise, and they'd just be like, after a few days, brick red, you know, from the dust coming off of Africa. So, in addition to our Crete samples here, which is very short transport distance for our Saharan dust, Bill Landing has been very generous with samples from Geotrace's cruise, taken more or less in the middle-ish of the Atlantic, and then he also has a Bermuda sampling site. And so looking at filters, and we also were able, from a Spanish colleague who had soil samples from all over the Sahara, Libya, Morocco, Tunisia, and various other places, we were able to aerosolize soils and characterize the iron composition. Not surprisingly, most of the iron in aerosolized soil is iron oxides. So that's mostly the starting material. And if we look at the aerosols coming from the Sahara in Crete, they're mostly iron oxides in the green, a little bit of iron silicates, a little bit of iron 2 sulfate. If we look at these samples taken aboard ship, now you can see that iron oxides are only about half of them, and we have iron 2 sulfates coming in, and we actually, in our fits, we're still scratching our head, we see a fair fraction of iron 3 phosphates in these samples taken out in the middle of the ocean. And then over in Bermuda, again, certainly lower percentage of iron oxides relative to Africa, and we see iron 2 and iron 3 sulfates. So it does seem like that the composition, there are reactions that are occurring from Sahara dust getting to these different locations. Now, of course, it's not ideal. These are not all in the same flow path and everything, but if we look at iron in various places in the North Africa, it's pretty much the same in composition. The other thing is when we do our bulk spectroscopy with the X-ray optics out, we can essentially use features of the spectra to characterize the oxidation state, bulk oxidation state of iron in our sample, in our 3 locations. And so iron 3 is delightfully in this reddish-orange color, and iron 2 is in the blue color here. Again, close to the source, mostly oxidized iron 3, or farther, we can see that we see much more iron 2 in the sample. So where's that coming from? So we speculate that that is some reflection potentially of photoreduction processes which have been suggested for aerosols, but you know, in terms of evidence in natural samples, there's a little, but not a whole lot. So, yeah. So one of the things Amelia did with high split trajectories and lots of calculations, she could come up with this sort of travel time variable versus the position of the pre-edge centroid, which is the thing that we use to tell iron oxidation state. And essentially, what we're showing here is at this side of things, it's a more reduced iron. On this side of the graph, it's a more oxidized iron. The longer something's in the atmosphere, by any measure, you've seen the reduced iron does seem to more or less grow in. For all the samples we ran on this the soluble ions have been measured, so the calcium, potassium, magnesium, sulfate, chloride, and that sort of thing with IC. And when you have that complete suite, you can put all that chemistry into the isoropria thermodynamic model developed by Thanos Nennis and calculate an aerosol pH. And I know a table's not the best thing, but if we look at Bermuda, which is our maximum travel time samples, and we do this pH calculation, we have numbers 3s and 2s. Generally, if we look at our geotraces and our Crete samples, the calculated pHs, except for one, are all in about the range of 7. There's assumptions of relative humidities, but no matter, you can use different ones, you still get this sort of two haves or have-nots. The Bermuda samples always seem like they are more acidic and relative to the other sets of samples, and maybe not surprisingly, the solubility of the Bermuda samples is much greater. So here we have the total iron in the sample. So these gray bars are Greece, the Crete sites. The middle 3 are the geotraces samples, and the dark bars are Bermuda. And this dotted line with the circles is the percent of soluble iron in the samples. And basically, the Bermuda samples are the most soluble and the most acidic, essentially, by our calculations. So we can, the take-home message from this part of the talk is, with the increased transport time in these 3 different sites, we see a growing in of iron 2 and a little bit of iron 3 sulfates in our aerosols. The iron oxidation state becomes more reduced with transport time, and it looks like the aerosol acidity increased. And all of those would likely lead to increases in the overall solubility of iron and almost likely phosphorus in these aerosols as well. So there you go. So that's what we've been doing with aerosols. Yeah, sure.
So on the phosphorus side, so the indeed there's a difference in the solubility, but what about the fluxes of European versus North African aerosols to the Eastern?
That's a great question, and I think we tried to figure that out, and I think we think that the maybe overall aerosol flux is maybe about this. Well, maybe I'm thinking of the flux. They may deliver the same amount of soluble phosphorus, so there's less of a flux of European aerosol, but you only more soluble, so you only need a third as much, but it has the same impact. So one could then, I think we tried to wave our arms because it was a Nature Communications paper, so you have to do this, like, over amplify, like, we've solved all the world's problems. Is that, you can find climate models that say anything, but that there may be, as with climate change, more of an influence of European winds. Some people predict for the Mediterranean, so.
And a second question on the last part is, so as you get across the Atlantic, my understanding is you can get increases in Asian and North American dust. I mean, it seems like a long way, but it's not just North Africa dominated, although maybe when these samples are Bermuda.
You're absolutely right, and we do worry a little bit in the Bermuda. There's, these high split back trajectories have their own problems, especially when you go long time scale. So we do have from Bill samples that both their trajectories and such identify them as being more North American sourced and more African sourced, and we can't use this in basically as a discriminator really in the paper, but the ones that that are African sourced are that reddish color, and North
Yeah,
America.
that's
So,
the diagnosis.
so we are working on the sort of big iron paper, as I call it, where we're going to try to integrate, because it was making our head explode, trying to integrate both differences. You maybe have starting differences in the composition of the North American dust versus,
That's true.
and
That's right.
and then try to integrate that. And then with the North American, you can add, if you have, let's say, some SO2 additions and then acid formation. It's so we have a lovely but slightly insane colleague at Argonne who has been running around getting aerosol samples from all over the world, Patagonia and India, and the list goes on for us to characterize, and we're going to try to put this all into some coherent thing to try to describe what the bigger factors are. Yeah.
So how would a spectrum of organophosphate fertilizer look compared to your bacteria samples? I mean, you
Well-
ascribe the organophosphates to microbes or bacteria at least or something, but what about fertilizers or not fertilizers? I guess they use them as fertilizers.
Oh, pesticides, we probably could not. It is a really, in terms of phosphorus, lousy tool for discriminating different types of organic molecules. That's just the bottom line. You need to do something else.
So you get this increasing solubility with time. Does the photochemistry just not work in surface soils? I mean, the question of how much of it has to be in aerosol, or if you have a very, like exposed, I'm thinking, Venezuela or whatever, that you have very high light impact in surface soils, would you still get solubilization of material or not?
I don't know. I mean, that's a good question. I mean, one of the other components, which we don't have, any evidence for, yay or nay, is that these photoreduction reactions, and you read the papers, they almost always have to have some involvement of some sort of organic ligand or something as part of the reaction. Those, again, you'd presume would be fairly abundant in soil organic matter. You got all sorts of organic molecules, but why you don't, the baking desert sands of the Sahara should be, reduced iron metal, probably. It's so sunny there.
So would you see, one way of looking at that would be if you saw it at the beginning of a dust storm versus the end. Presumably the end stuff is coming deeper.
Maybe, yeah.
I don't know anything about aerosols.
I have a question.
Okay.
Do you want?
Yeah, well,
I'm
do
not
you have any
commenting.
thoughts about that?
Sorry. Regarding sampling, if you sample from the ship or along the track, is that size discriminated, or you sample everything PM 2.5?
I think these are PM 2.5. So.
So then to follow up on curves, because I guess you sample then all the other particles. You have sea salt, you have sulfuric acid associated with sea salt, you have organic material. I'm wondering, I could imagine what you showed, that the dust dominates. It depends. If you're in the plume, yes. If not, maybe you have some sulfate from sea salt, which helps.
Yeah.
In principle, it doesn't matter in that sense, but it's maybe not associated necessarily
With
with
the-
iron, but
Yeah,
then you have.
You can't tell. Yeah, you don't. But it's.
On a single particle base, if you would go particle by particle, you could, with your resolution, you could say that's a mineral dust, it's not a sea salt or so, but then you have less signal.
Right. I mean, and on the individual particles, that does help, because how you would form an iron phosphate. I mean, the ocean waters, don't have a whole lot of phosphate, and so you would need to probably have a particle containing maybe a little apatite, a little iron, and a little acid.
Yeah, it was not for the source, but just to make it more soluble, that you have sulfates or some oxidation potential, or you have organic acids, which could then from the sea salt or sea gray aerosol.
Yeah.
React. That was just not from the source of phosphorus or iron.
Right, but I think
to increase your solubility. And one more. If I
Yeah,
may.
go.
I really love that you do the standards, that
Yeah.
you measure that through.
Yeah.
You try something similar. You have it for the phosphor edge. It would be very neat to extend it for the carbon edge. That's first of all, because we are working on the carbon edge.
Yes.
Working on the carbon edge, and of course now with mineral dust is a big ice nuclei, and so your standards are really great for that. But then, of course, the mineral is maybe associated with organic material like humic acid, soil humic acid, and there you need the carbon edge. And I just saw your standards, and it would be great to combine different KI.
I would totally agree that, I mean, we, but, you know, as you know, carbon is like, I think they have a special level of hell reserved for people trying to do synchrotron carbon. So, oh, I'm sorry. Did I? or do you agree? I mean, it is very difficult because the beam.
It's not quite helpful. Purgatory.
But nevertheless, you know, challenging can also mean really worthwhile because obviously a lot of people, for some reason, are really concerned about carbon cycling. You know, so I mean, yeah, if you can do that, that would, you know, be awesome and make it available for
Maybe
folks.
you should write a beamline proposal for your mineral dust samples. Just give it a shot.
For carbon.
Yeah, for carbon, but then align it with your knowledge about how much phosphor and iron in it. You know, same samples, and then.
So the only thing that, you know, since you'd have to get the aerosols off the filter matrix because it's a, you know, Teflon which contains carbon. So
We sample directly on the substrates.
yeah. So
Yeah, we put the substrates on the impactor stage, and
that's.
Okay,
But that, yeah, well, but I mean, we have lots of, you know, we could work this out, you know, but that'd be very worthwhile, but very challenging. But, you know,
Well, two things. The first one is sort of going along with Mary's question. So how, do you have any idea of how high these aerosols are getting up in the atmosphere, or where, like, what's going on? You know, once they leave the ground, what's happening to them?
I guess for our back trajectories, we do pretty much have to make a guess that, you know, that they're fairly high up, you know, 1,000 meters or more in the atmosphere. I think I can't remember the specifics that Amelia was using, but so, you know, they have to be probably fairly high up to get transported. Those kind of
transport 2 to 3 kilometers.
Right.
Yeah.
And so, like my idea is that, well, are they exposed to significantly more UV radiation up there? And that maybe is why the photo reduction is happening up there and not, you know, on the ground surface or something.
Oh,
like that.
yeah, it's maybe a thought that, yeah, I mean, yeah.
we come from some place where there's no cloud interfering. They're just bombarded all the time anyway.
No, but there is aerosol photochemistry. That's relatively new. It's funny that they, in principle, rediscovered the marine chemistry. So on interfaces, on solid liquid interfaces, and then you have, how do they call it, photosynthesizers. That could be iron or humic acids, even organic molecules, and they can then trigger reactions. But it's I think for marine people, they would be, I'm not laughing, but the aerosol people rediscovered that, and I think Thanos Nenes thought long also about that, or has some ideas, but that's out there, but it's not yet so established, and the experiments are tough because it's not bulk, you know, you have a nanometer-sized particle on which you have to study interfacial photochemistry, so it's on a completely different level what you usually do in marine or in normal chemistry. You don't have the samples when you work with aerosols. So in the moment there are only 3 groups, I think, who try that. Yeah, so it's tricky to do it on the submicrometer scale.
Sort of more along that, I guess, and then does the weather patterns there permit that there's a lot of, you know, moisture in the air? Because you said one of the things that happens or a problem is that if you get moisture with your particles, then yes, they can react with things. But well, I'm thinking like sort of an acid rain. So when you're producing acid rain, you have, you know, sulfuric acid forming, in the dust particle, or in the water aerosol particles, and then would that interact with these dust particles, and then?
Yeah, I mean that's another factor. It may be certainly much we would suspect more relevant for North American sourced aerosols where you do have SO2 inputs from various power plants and other exhaust kind of things that could definitely impact the solubility. So.
And sorry, one last
No,
thing.
it's not like there's a charge for each question.
That's what you think.
Well, my memory could be completely off here, but one of the things that I think I remember reading about, especially in Jamaica, was that they did have a problem with people coming down, or especially young children coming down with respiratory illnesses. And one of those big things was because of the dust storms coming from Sahara. And I can't remember if it was because of the dust itself or actually because of bacteria associated with them getting in their lungs. And so if you haven't heard about that, or I'm actually interested now in going to look back at that to see if they thought it was just the dust or bacteria associated.
No, I think that the bacteria is a hot. I mean, people that are using molecular techniques are trying to do a much more elegant detection of bacteria than we can do. Like, whoa, there's a blob of phosphorus containing something or other, and that might be a bacteria, or like these sulfur blobs that we're seeing in the sulfur zero in Atlanta aerosols. I go to every atmospheric colleague in our department, we have quite a few, and I say, oh, I got these particles with sulfur zero in them. Where's it coming from? And they go, huh? You mean organosulfates? No, not sulfates, sulfur zero. Huh? So nobody has an answer for me. So, and I think, well, what thing would have sulfur in a relatively neutral oxidation state, and when we look at our sulfur, they look like globs, little submicron globs. Well, there's a lot of, organosulfur, sulfur-containing amino acids with sulfur in the approximately zero oxidation state. Maybe bacteria, again, it's arm waving, but, I think smarter folks are going with molecular biology to really try to do what's there, because I think that'll be really interesting for a number of reasons, health or just transport of stuff.
So is fluorine in your analytical window? I mean, it wouldn't be on a Teflon, but are you able to resolve fluorine fluoride?
Let's see. I'm trying to think. I don't recall. Certainly not on our low energy line. It's above the absorption for that. And I don't think we've, for whatever reason, maybe because we have almost everything we have is on Teflon, we probably just don't ever look at, we turn the fluorine map off because it's just going to be not very interesting. But I can, it should be possible.
I think that would be a really, really interesting one to couple with iron and phosphorus, and get on marine versus different mineral sources, because there's a lot of fluorine chemistry associated with, different mineral reactions. So.
Yeah. Well, I'm gonna on my little table.
Not with Teflon.
No, obviously not. But I'll just, it's one of those lower mass elements, and those, as you get lower and lower, like carbon. Oh, never do that.
Well, can't you use different substrates?
Yeah, you can use different substrates, but it's just the signals and essentially the bang for your buck of photons hitting your sample. The lighter and lighter the target, the less signal you get, and the less penetration the beam has. So it gets more and more difficult. But I think fluorine should be possible. I just need to look into that. I don't know.
Okay.
Okay, we're going to give you a little break now. before you're going to go
Alright.
because you're going to go back into the water.
Yeah, yeah, but we'll take you
Mine's
back to
very
aerosols
simple.
later in the
Too
day.
bad, no,
You have
they-
aerosols
Yeah,
at
thank you.
4.
Thanks.
So
Thanks so much.
Oh, thank you.
Tomorrow we'll fill the room for you.
This is awesome.
And then stuff you.
Thanks for giving us
Oh,
really
my
appreciate
pleasure.
it.
Love your questions.
Yeah, I have to rush to class. I have 3 minutes afterwards. So I see you in my lab, I guess.